In the name of Allah, who is the most merciful and most compassionate, welcome to my new video. Today I am going to talk about a very important topic, that is apoptosis. In this lecture, I am going to explain the definition of apoptosis, the differences between apoptosis and necrosis, and the pathways occur during apoptosis. Let's start our lecture. Apoptosis is a type of cell death which is programmed or suicide. Why we call this suicide or programmed? Because in this cell death, all the process is done by the cell's machinery. Enzymes are activated and the rest pathway is done by the enzymes of the cell. For this reason, we call this programmed or suicide. Differences between necrosis and apoptosis. Necrosis is happen just pathologically because of external injuries, but apoptosis is happen pathologically and physiologically. For example, during embryogenesis, some parts of our body are disappeared. It is a physiological action, but sometimes it is happen pathologically as well. Membrane integrity. Necrosis, necrotic cells lose its membrane integrity at early stage. For this reason, all the enzymes, all the components are leaked out. So enzymatic digestion is happen. After that, inflammatory process will start. But in apoptosis, apoptotic cells maintain its membrane integrity until late stage. They don't let its components to leak out. At late stage, these components bud out like a package. All these components are inside this package. We call this package apoptotic body. After that, cell releases some molecules on the surface of these apoptotic bodies. We call these molecules opsonins, which attract the macrophages to come and phagocyte these apoptotic bodies. So we can say no enzymatic digestion, no inflammatory process. Mechanisms of apoptosis. Apoptosis is happened by two pathways. First one, extrinsic or death receptor pathway. Second, intrinsic or mitochondrial pathway. First, I am going to explain extrinsic pathway. For example, this is a cell. It has some death receptors, or we call these receptors FAS. Means first apoptosis signals. First apoptosis signal receptors, or TNF, tumor necrosis factor. These receptors are activated by some fast ligand. Ligand is used for those things which bind receptors and activate them. These receptors have some dead domain. Why we call this dead domain? Because it binds with other proteins which are involved in cell death. These dead domains activate adopter protein which also have some dead domain. These dead domains activate initiator caspase, which is number 8. We have some types of caspases. For example, initiator caspases and executioner caspases. Initiator caspases are those caspases which are activated at early stage of apoptosis, but executioner caspases are activated after initiator caspases. This initiator caspase number 8 activates executioner caspase. Now, what is the function of executioner caspase? It degrades the components of cytoskeleton and nuclear matrix, or we can say it digests the proteins which are the components of the cytoskeleton and nuclear matrix. And also, executioner caspase activates DNase. After that, there is a blip. Blip is made. This blip consists of genetic materials, proteins, enzymes, which buds out. We call this structure, this package, apoptotic body. After that, cell releases some molecules on the surface of these apoptotic bodies. We call these molecules opsonins, which attract the macrophages to phagocyte all these apoptotic bodies. So the apoptosis process will finish. For this reason, we can say there is no enzymatic digestion. No enzymatic digestion, no inflammatory process. Second one is intrinsic pathway or mitochondrial pathway. Why we call this mitochondrial pathway? Because inside the mitochondria, there are some poisoners. There are some deadly molecules. For example, cytochromicin and apoptosis inducing factors. Whenever they go out, the apoptosis process will start. Now I want to share one thing. The survival of the cells refer to two genes, pro-apoptotic genes and anti-apoptotic genes. Pro-apoptotic genes are bad, and also there is bad, but bad is for survival. Anti-apoptotic genes are BCL2 and BCLXL. Now the question is that how these genes determine the survival of the cell or how these genes are activated. Whenever growth factors, hormones, surviving factors stimulate the cell and give signal inside the cell, these signals inhibit the pro-apoptotic genes, hold the pro-apoptotic genes, and stimulate or express the anti-apoptotic genes. Anti-apoptotic genes make some dammers to close these tunnels, to close these spores of mitochondria and inhibit the escaping of cytochromacy in AIF. And also BCL2 inhibits some apoptosis activating factors. For this reason, we can say the cell survives for a long time. When there are no growth factors, hormones, and surviving factors, the anti-apoptotic genes will be inhibited because there is no stimulation for anti-apoptotic genes. But pro-apoptotic genes will be free. Pro-apoptotic genes produce some products which open these tunnels, these pores. Finally, cytochromacy and apoptosis inducing factors will go out. Apoptosis inducing factors bind anti-apoptotic factors and neutralize all these anti-apoptotic factors. But cytochromacy binds apoptosis activating factors. Both of them activate initiator cascade, which is number 9. You know initiator cascade activates executioner cascade, then executioner cascade does its function. You know, bilibismate, macrophages will come and phagocyte all these components. This is the intrinsic pathway of apoptosis. And also there is another pathway which is called perforin grains MP pathway. In this pathway, cytotoxic T cells release some granules which consist of perforin and grains MP. Perforin make a pore for grains MP to enter the target cells. What is the function of grains MP? Grains MP activates executioner cascade, and after that executioner cascade does its function. The rest pathway is the same. This was our today's topic. For more medical lectures, please subscribe our channel. Thanks for watching.